So thank you all for coming back promptly, so we stay sort of on time. We now are going to transition to our fifth and last session of the workshop, which is to discuss the issues around clinical investigations related to MRT. And our colleague Jeff Botkin is going to chair this session. Jeff.
Okay, thanks, Jeff. So this is clinical investigations. We're going to have a couple of objectives here. Discuss the preclinical evidence base necessary to support a first-in-human MRT. Consider the earlier precedents for collection of safety and efficacy information for novel techniques, and then to discuss what an ethical clinical investigation of MRT might look like. So our first speaker, under a section called Preclinical Evidence Base to Support an IND for MRT, is Liang, who is a preclinical reviewer in the Office of Cellular, Tissue, and Gene Therapies at the FDA. So welcome.
Thank you, and good morning. I will start with the regulatory expectations for mitochondrial manipulation technologies from preclinical perspective. So first, I will have a general introduction for the preclinical testing concept. What are the objectives of preclinical testing? The preclinical testing is to support the safety and provide the scientific basis of the administration of investigational product in the target patient population. And preclinical testing is to provide evidence of the acceptable benefit-risk profile. And preclinical testing is to inform the design of the proposed clinical study. This may include to support the enrollment of appropriate patient population and to recommend a safe starting dose and dosing regimen, and to guide adequate clinical monitoring plan and stopping rules. So preclinical data is an important element for IND application. What regulations govern preclinical testing? This slide is a selection from the IND regulations from Chapter 21 of the CFR. The regulation states that prior to a clinical trial, a sponsor should provide adequate information about the pharmacological and toxicological studies. The kind, duration, and scope of animal and other tests required varies with the duration and nature of the proposed clinical investigations. And for preclinical testing strategy for cellular and gene therapies, we have a guidance document which was published about 1.5 years ago, Preclinical Assessment of Investigational Cellular and Gene Therapy Products. This guidance document clarifies our current expectations for preclinical data that would support the IND and BLA application. In the next two slides, I will have a high-level summary of the preclinical testing program. So preclinical testing program should be stepwise and science-based. This will include proof of concept or POC studies and small pilot studies and also definitive studies. Data generated from the proof of concept studies can support the feasibility and activity of the product and associated study procedures for specific indication. Sometimes smaller pilot studies that will be conducted, the data from pilot studies can explore key parameters to guide definitive preclinical study design. And the definitive safety studies are the key IND-enabling studies that are conducted to support the proposed clinical trial. So preclinical study design considerations, these are very general considerations. Use of relevant animal species and models. And remember to apply the 3 R's: reduce, refine, and replace. And the study design should be non-biased. This can be achieved by animal randomization and blinded assessment. adequate numbers of animals should be included in each group, and the study design should mimic the proposed clinical trial design as closely as possible. And the study should include adequate safety and/or activity and endpoints. The study duration should be sufficient to allow to assess the applicable endpoints. Next, I will move to the preclinical consideration for mitochondrial manipulation technologies. Last year, we held an advisory committee meeting to discuss oocyte and embryo modification to prevent transmission of mitochondrial disease. In the next several slides, I will summarize the committee's discussion on preclinical issues. The committee was asked to discuss available animal models and in vitro methods to address the safety and prospect of benefit of mitochondrial manipulation technologies. These are the preclinical issues raised by FDA CBER. The concerns include the possibility of inadvertent damage to the manipulated oocyte or embryo, the long-term risks associated with the carryover of abnormal mitochondrial DNA to the offspring, the potential for abnormal embryo fetal growth, resulting in offspring with significant defects. This slide highlights some general considerations cited by the committee members. There is not sufficient animal data to support the use of mitochondrial manipulation technologies in first-in-human clinical trials. The committee members recommended continuing studies, continuing in vitro studies, and also studies in animals. And for the studies in animals, multiple animal species models will probably be necessary to assess overall safety concerns. Sufficient numbers of animals, including both mothers and offspring, are needed to adequately evaluate the safety concerns. And long-term follow-up of offspring throughout of developmental stages will be necessary with possible multi-generational evaluation. So prior to the clinical investigation, clinical data is required. This is to meet the regulatory requirement. And what type of preclinical data will be required for a specific clinical investigation? Well, our guidance document can provide some general information for the preclinical testing program in the for the products that are regulated in our office, OCTGT, are highly individualized. So we encourage the sponsors to have early interaction with OCTGT. And the device can be achieved by pre-IND interactions and pre-IND meetings. Pre-IND interactions are non-binding, informal, scientific discussions between OCTGT non-clinical review disciplines, meaning farm talks and CMC with the sponsor. And pre-IND interaction is about initial targeted discussion of specific issues. And pre-IND meetings are non-binding, formal scientific discussion with clinical and non-clinical review disciplines. And for pre-IND meetings, FDA will generate meeting minutes. The pre-IND meeting package should include summary, data, and sound scientific principles to support use of a specific product in a specific patient population. And here's some contact information for OCTGT. And I will stop here. Thank you very much.
Thank you. Appreciate it.